Hi, everyone, and welcome. My name is Katie Sacra, and I am the Director of Family and Community Engagement here at the GFPD. I will be moderating today's webinar alongside Ryan Maple, our Executive Director here at the GFPD. We are so glad that you're able to join us for today's event, From Genes to Hope, Unlocking the Power of Genetic Information. We know genetics can feel overwhelming, especially when it comes to a rare disease, but understanding the basics gives families power to ask questions, to participate in research, and to help move science forward. This webinar is not about memorizing science, and hopefully it won't feel too much like you're back in biology class. Instead, it's about understanding just enough to feel confident, informed, and included in these important conversations and decisions that rare disease families often face. To help make that happen, we are so grateful to have Dr. Nancy Braverman here with us today. Dr. Nancy Braverman received her undergraduate degree from Cornell University and a Master of Science in Genetic Counseling from Sarah Lawrence College. She received her Doctor of Medicine from Tulane University and completed a pediatric residency at Yale University, followed by a fellowship in medical genetics at Johns Hopkins University, where she became involved in peroxisome disorders as a clinician scientist. She has continued this important work throughout her career, combining clinical care with research to advance our understanding of these rare diseases. Dr. Braverman is a professor in the departments of human genetics and pediatrics at McGill University and a senior scientist at the Research Institute of the McGill University Health Center. She is also an attending medical geneticist at the McGill University Health Center, and she serves on the Medical and Scientific Advisory Board for the Global Foundation for Peroxisomal Disorders. After her presentation, she will take some time to answer your questions. You're encouraged to submit questions at any point using the Q&A button or the chat. During her presentation, Dr. Braverman will be introducing and explaining many key terms, but if you hear something that's unclear or you'd like to understand a bit more, this is your chance to ask. We're here to help you feel empowered to ask questions, make informed decisions, and take part in the conversation. So please share your questions as they come up, and we're going to take time to discuss them in just a bit. Dr. Braverman, thank you again for being here with us today, and I'm going to turn it over to you.
Thank you, Katie. I'm really glad to give this presentation. I hope it's helpful, and thank you all for coming. So we're going to start this talk from the very beginning, getting the diagnosis of an infant or a child or an adult with Zellweger spectrum disorder, and thinking, where did that come from? Because no one in our family has this. And so this slide shows a 3-generation family history with the affected child shown by the arrow. And then there's an older sister who's 2 and an older brother who's 5, and then the parents. And this also shows the parents, brothers and sisters and their children, as well as the paternal grandparents and the maternal grandparents. And no one has this disorder. So it does all make sense when we realize that we're all carriers for genes that don't work right. And so if you were going to go ahead and test these family members, you would find out that the child who's affected has 2 disease genes for this trait, which is how this occurs. And he got one of them, which is represented by the small a from each parent. So the parents are carriers of one gene that doesn't work right for this trait, the small a, and one gene that is normal for this trait, which is the big A. And if you move forward in the family history, they got the gene, this small a, from one of their parents each, and one of their parents, who was the carrier, then passed it on to other children and to their children. But nobody has this disease because they're all carriers. So the disease presents itself in a family when 2 carriers have children together who are carriers for the same gene that doesn't work right. So, and then there's a chance that the child inherits both small a's and has the disorder. So this type of inheritance pattern is called autosomal recessive inheritance, and this is just a chart that you may have come across when you were in school. But here is the father who's a carrier, so he has one gene that works correctly for this trait, which is the check, and one gene that doesn't work, which is the X, and then mom, who's also a carrier, and she has a non-working gene for the same trait, and it's in the same gene. And so when they have children together, they can either pass on the gene that works correctly or the gene that does not. So here there's one chance that they pass on both genes that work correctly and the child is unaffected. Or there's two chances, this one here and this one here, that they pass on one working and one non-working copy or a carrier like the parents, or this chance, this working copy and this non-working copy. So they're also a carrier like the parents. So you could say that 2 out of a total of 4 times the children are unaffected but are carriers. And then there's 1 out of 4 or 25% of a chance that the child could inherit both of the non-working copies and therefore be affected. But the way we like to explain this to families is that 3/4 of the children will not have Zellweger spectrum disorder because carriers are unaffected and we're all carriers. So it doesn't matter whether you're a carrier or you're not a carrier, you're normal or you're normal for that trait. And there's only a 25% chance in each pregnancy that the child would inherit both of the non-working copies and be affected. So as I mentioned before, we're all carriers of several pathogenic variants in genes that cause recessive disorders. We pass on 50% of our genes to our children, and we have little control over which genes we pass. But remember that our children, in addition to getting a disease gene, are also inheriting 30,000 other genes that can modify the disease picture. And this is why we're all unique individuals. And this is why even if you have the same gene changes, you have 2 children in the same family who have the same gene changes, they might have a different expression of the disease because it's being modified by all these other genes that they've inherited as well. So we're all unique individuals. And knowing what gene changes are causing the ZSD in your child is empowering. As you heard Katie speak, this is really going to be the essence of what this lecture is about, how knowing how to interpret these changes and what they mean will be useful to you. So subsequent to receiving a genetic diagnosis, there's often relief of knowing what the disease is and moving forward based on the knowledge that we have about that disease. Um, you're able to predict which relatives could be at risk because remember the family tree, we know who's a carrier. So if a carrier has children with another person that's a carrier, then they would have a risk to have a child with ZSD. If they have children with a person who's not a carrier, there's no risk because there has to be 2 genes that don't work right that the child inherits. And so you could also do family planning with this. And then a treatment or management might be available, and many times it might be available targeted to what the gene is that's involved in Zellweger spectrum disorder or what the mutation is in that gene. And then also subsequent to a genetic diagnosis, at a research level, you could take it to the level of figuring out how that change causes disease. And in many ways, families have, by doing this in the past, they've enabled us to understand what gene changes cause what protein changes, cause what function changes. And this all leads to new treatment because the more we know about this pathway of gene change to disease, the more we can figure out ideas of how to intervene and then modify that. So the terminology that we use in genetics that you'll hear about often is pathogenic variant, and that used to be called mutation, and it's a change in the DNA. And the change has been sort of moved over to variant now because it's more acceptable at the level of the fact that it's just a variant change because it's inclusive of all variants and many variants are harmless. So this is just another variant. And then we classify that variant as to whether it's pathogenic or not. So we're trying to move away from the term mutation. It's been difficult because it's ingrained in the literature, but we're trying to move away to variant and then what type of variant is that? So you'll hear pathogenic variant instead of disease causing variant, gene variant instead of gene mutation. And then the term that you probably have all heard of, and that we'll tackle a bit in this lecture, is a variant of unknown significance rather than an uncertain mutation, meaning this particular variant, we're not sure what it means. We're not sure if it's benign, and we're not sure if it's pathogenic. We're not sure if it's causing the disease in this individual. So, in giving analogies to genes and variants and RNA and protein, a good way to think about it is your body's instruction library. So you have the person who's the library, and then chromosomes are the books in the library, and then genes are the chapters in the books, and DNA is actually the sentences in the chapter. But they're made up of base pairs, of which there's A, T, C, and G, so only 4 base pairs, or the letters in the sentences. And then a mutation or variant should be a typo in the DNA letter. And then RNA is a photocopy of that DNA. And protein would be what the meaning is of that sentence or product. And then expression is actually reading the chapter. What does it all mean? What does it all translate into? So I'll show you this in more detail. So when the letters change or are missing, this is when you have variants that occur. So here's a sentence that says, the cat ate the rat. That would be the sentence you want. But now you have a variant, and instead it's the cat ate the bat. So there's going to be a different meaning in the end for that sentence. And that we call a missense variant. So the sentence is still the same. The spaces are still the same between the sentence, but the word has changed. And so the meaning in the end has changed. This is a missense variant. The sentence is still there. The meaning has changed. The other example is for the cat ate the rat. And now we have a deletion of that C. And since these are 3-letter words, you can see that that's changing the whole frame. If we remove that C, that's the variant, then we have the ATA, TET, HER, AT. So that's called a frame shift. We've just shifted the whole reading frame by deleting one residue. And this ends up being nonsense. We've just changed the whole sentence. It's nonsense. It doesn't mean anything. And in sort of the rules of the way proteins are made, this is sensed and this protein is not made. This is protein is gone because it doesn't code for anything meaningful, and the cell decides not to even make it. So again, removing the first C scrambles the entire sentence, and this will be a major effect on the DNA, whereas example 1 is kind of a minor effect because the sentence is still there, and it kind of makes sense, but it's not completely the same as the first sentence. Whereas in the second example, we've changed the whole frame, it's nonsense, and it's not even made at the protein level. So changing the space between the letters can also have a major effect, and these are called splice site variants, in case any of you are familiar with those. And for splice site variants, usually they change the frame also, and they're deleterious. So here we go back to chromosomes. It will be the book containing the genes. And so as I mentioned before, we get half of our genes from each parent. And genes are on the chromosome, so we get half of our chromosomes from each parent. So you could see here, and the chromosomes are arranged in pairs from 1 to 22, and then you have the sex chromosomes X or Y. And so you get one chromosome of each pair from each parent, so one from the mother and one from the father. And so your genes are on these chromosomes, and this is how you get half your genes from your parents, because they're coming from half your chromosomes. So let's go specifically to the PEX1 gene. So we know its location. It's on chromosome 7, position Q2.1.2. So that's like, just to go back to the slide, that's like the map, the reading map. So if we go to chromosome 7, so at position Q21.2, which is sort of on the lower portion of this chromosome, but these chromosomes are not shown in bands, so I can't find the exact sort of GPS location on that chromosome, but that terminology tells you exactly where on chromosome 7 PEX1 is. So we know it's on chromosome 7, and then we have all these numbers here. And so the number on top is the number at the genomic level. So this is going to include all of the sentence as well as the spaces in between being numbered. So it's always a big number, and you usually don't see this number, but you do see this number, which is the C number. And the C number refers to starting at the first base pair of DNA of this gene that encodes protein is 1. And then all the way at the end, these are in 10 intervals, 10, 20, 30, 40, 50, 60. This is 60. So C60 is right over here. C120 is over here. C180 is over here, and so forth. And these are the DNA letters, the A, G, C, and T, that are going to be arranged in a certain way that code for amino acids, which are underneath. These are the single letters for amino acids. There's also a triplet for amino acids. So this is methionine, also called MET, tryptophan, glycine, serine, aspartate. I know this because I've been doing it for so many years, but this is what this translates into a specific amino acid, and you can look that up. And so this is at the P level, or the protein. And so this is at the end here. There are 60 base pairs, 3 base pairs encode an amino acid, so there'll be 20 amino acids here. And so this is how you read this. So here's amino acid 40, here's amino acid 60, here's amino acid 80. So the G, as I mentioned before, is a sequence of the entire gene, including the spaces. The C is a DNA sequence of the part that makes the protein, and it's the letters A, G, C, T in 3-letter codons that code for the protein sequence, which is the amino acids. And just to go back to sort of the location, because that's important too, the DNA is in the nucleus on the chromosomes, and the RNA is the DNA photocopy that travels to the cytoplasm. And then the protein is actually made from this photocopy in the cytoplasm and goes to the places it's supposed to, in this case, the peroxisome. So if we want to figure out what the gene change is in an individual, and we get a report back that says it is the PEX1 G843D change, and I'm using this as an example because it's one of the more common changes. So many of you might be familiar with it. And so the protein is sort of the workhorse of the cell. So we kind of like sort of focus always on what the changes do to the protein. So protein is a glycine at position 843 of the protein is changed to aspartate. And I'll show you that on this map. And it's in PEX1. And what you'll see in your gene report is a DNA change. So what's the change of the DNA letters? That's the C number, right? So at 25, position 25, 28, there's a G to A change. So let's go take a look at that. Here on PEX1, this is position C2520. So now, and we know that this is C2530 because it's every 10. So if we go back to, then we have this. We'll have to go 1, 2, 3, 4, 5, 6, 7, 8. So this G here is changed to an A. So that's this nomenclature, C2528, G8, tells us exactly where that change is. And then we know that what that change does is it changes this glycine. This is the amino acid underneath it. It changes this glycine to aspartate because now the code, instead of this GGT code, is a GAT code, and that changes the glycine to the aspartate. So the change at the DNA level makes a change at the protein level. That's being encoded. And that was the missense variant that we showed earlier, the example 1. So the protein's still made. It kind of has the same sense, but it's a little bit different because of that one G to D change. And that actually, even though it looks a little bit different to us, it actually causes a big difference in the function of the protein. And so once we make this protein now, it actually has to assemble into something that functions. And the PEX1 protein assembles with with five other subunits. So six subunits of protein assemble in this kind of sort of propeller structure here. And there's three PEX 1s, they're in light gray, assembled together with three PEX 6s. So if this protein, if this G change at position 843 in the protein changes to an aspartate, what happens is that change happens to be on the interface between the PEX 1 and PEX 6 residues, so they don't assemble as well together. And that's the functional effect or the functional expression of this change. And so now we can't get PEX 1 and PEX 6 to get together in this six-membered structure exactly like this. And therefore, they get together in some sort of structure, but it's not as stable as this. It degrades more rapidly and it just doesn't function as well. But it has some function because we know that patients who have this change are not as severely affected as patients who have changes that result in no protein being made. So, just in summary of the different kinds of pathogenic variants, we talked about the missense. So that's like PEX1 G843D. We have a change of an amino acid, the protein's made, but it's not functioning as well because it's just not folding, it's not doing what it's supposed to be doing as well. But it might be doing something. And then there are these other types of changes. This is in the nomenclature that we use in genetics called early termination change. And that's a change that would, at the DNA level, puts an early stop in. So there's actually a code for like the period at the end of the sentence, which means stop. And sometimes a change could put that stop all the way up at the front of the protein. That's called an early termination. You would make no protein, and that would be a severe effect on the protein function. And example two was a deletion, and the insertion's the same. You switch the frame, and often no protein is made. And a splice site, the spaces in between, when there's a change in the rules that govern those spaces, those also are usually severe and lead to no protein being made. So just moving on, there's not only one gene involved in Zellweger spectrum disorders, there's 13 PEX genes. And these protein products of these PEX genes all work together in a coordinated fashion. Not only PEX 1 and PEX 6, but they all do. And so defects in any one of them, I'm just going to go through this functional cycle for you so that you can appreciate that even though the genes are different, the patient's clinical pictures are often similar and overlapping because in the end, the peroxisomes are not functioning well. So the proteins aren't working, but they're all part of the same pathway. And in the end, you have less peroxisomes that are not working well. And so we see these similar features and we call them, we're all part of the same condition, the Zellweger spectrum disorders. And so you have PEX5, that's important in binding enzymes to take them to the peroxisome. So here we're traveling to the peroxisome. We dock at PEX 13 and 14. PEX5 slips through the membrane and releases those enzymes inside. And then we have PEX 2, 10, and 12 that then put a little handle on PEX 5 so that using that handle, PEX 1, 6, and 26 actually pull PEX 5 back out of the membrane. It's called receptor recycling and recycling it for another round of import. So you can have specific effects on the protein with these DNA mutations, but in the end, you're going to affect this everyone, all of the genes involved, defects will affect this assembly of a functional peroxisome. So the end result will be reduced numbers of peroxisomes that are not functioning well. And so you have a similar and overlapping clinical feature, although you do have, as we're learning, as we're seeing more and more patients with changes in different genes, which you're telling us because you found those changes, we see that there are some things, some things that are specific to certain changes and not in certain genes and not others. So this is a learning process. So, just going back to the common PEX1 variants, because you've heard us talk about PEX1 G843D here, that is very common. There's another one that's very common too. It's called I700 frame shift, and then this star is stopping. I have an early stop at amino acid 42 amino acids later. So this is going to be a frame shift. It's due to a deletion of a single base pair change, and no protein is made. As we mentioned before, this has a severe effect on the protein, and this is our missense allele. Both of these are very common, and they're common because they actually spread from Northern European ancestry. And they're common in North America because many of our ancestors are from Northern Europe. They may not be the most common mutations if you move to other countries. This is a spread that you can put together. This was put together by Karen Malone in 2025, looking at the public databases for variants carried in healthy individuals. So they looked at the I700 frameshift variant, all the carriers, and the G843D variant, all the carriers, and they could actually see from the databases are from different countries, they could actually see that this change was seemed to have its highest frequency in Scandinavia and then radiates out in a more diluted fashion. And then the G843D has its highest change really in Northern Europe, and then it's spreading out further south. And so I just thought this was sort of a nice map to show you how, these changes come from our ancestors and they come from certain regions where they must have originally occurred, and they're passed on from generation to generation. So if we move to the I700 frame shift, that's also very common. This is the protein terminology we talked about. And so the terminology at the C level, which would be the DNA encoding the protein, is that at the cDNA level 2097, and we'll go and take a look at this in a second, we have two T's instead of one T. That's going to shift the whole thing over in the frame shift that we showed before in example 2. And you have, because you're encoding nonsense, you get to a stop. And it stops 42 amino acids. And that protein we know from multiple lines of evidence doesn't exist. The RNA isn't even made. The photocopy isn't even made. And so common variants plus knowledge of protein structure and function enables us also to predict severity classes. So we have enough patients with PEX1 G843D and enough patients with PEX1 I700 frameshift to see these combinations over and over again, at least in North America and in Northern Europe. And so we can say that with some confidence that most of the time when you have two of these I700 frameshift alleles that encode no protein, you have a severe form of Zellweger spectrum disorder. And when you see a combination of one I700 frameshift with a G843D, you have a PEX1 protein with reduced function and intermediate, a clinical picture of intermediate Zellweger spectrum that's coming from this one G843D protein, because we know the I700 protein isn't made. And when you have two G843D proteins, you have more PEX1 protein amounts and you have a milder expression of the disease. So this is kind of like knowing the genotype, we can actually have some prediction of the clinical picture or the phenotype because we know about the structure and function and we've seen enough of the patients to be able to feel comfortable in this prediction. It doesn't mean it always happens this way because everybody's unique, but it gives us some ability to make a confident prediction. So moving on to what is a variant of unknown significance. So this means that the DNA was sequenced and they came across a variant in a PEX gene, but they're not sure that it's actually the disease-causing variant. So there's not enough data to determine if it's a disease-causing or meaning pathogenic variant or not. And I'll show you what kind of data we use to classify that. And it's the laboratory that's doing the gene sequencing that's going to make this classification, not the physician. So, but the laboratory will classify every variant that they find in disease genes as pathogenic, which means there's plenty of data to say this is disease causing, like the G843G and the I700 frameshift. Or it's likely pathogenic, means it's got enough data to make us feel that it's pretty likely the variant that's causing the disease in the patient. Or we don't have enough data to go either way, so we're calling it a variant of unknown significance, or we have enough data to go the other way, that it's benign, or it's likely benign, because remember, we all carry many, many variants, and not all of them are going to change the protein to the extent that it affects the protein's function in a deleterious way. And I kind of like to think of it as sort of a hourglass where you have, sort of this gradation that goes from pathogenic, likely pathogenic, VUS gets stuck in the middle, and then you have your likely benign and benign. So the criteria for classifying variants as pathogenic is it documented in unrelated individuals with a disease? This is one of the criteria. And this also tells us how important it is to put your gene variants into registries that people can see. So that because a lot of times the reason that the DNA diagnostic lab calls it a variant of unknown significance is because they've never seen it before. So they can't be, and they have to be completely sure. So they can't be completely sure that it's the one causing the disease because they never saw it before in anybody else. So the more knowledge, if we find, I think the number is like up to 5. We might need 4 or 5 other persons with that same gene change, and then it could be reclassified because we know that that change occurs in other individuals who have the same disease who aren't related to each other. That's very, very strong as disease causing. Another really strong line of evidence is how common is the variant in the population. So the variant has to be pretty rare for it to be considered a disease-causing variant. Because if it was common, then everyone would be a carrier, and then you'd have so many more cases of disease than you actually have out there. So variants that are frequent in the population are often not suspicious to be disease-causing variants. And then they also use lines of computational data. And these are multiple. And these will involve uh, things that are sort of AI-ish. So they're based on knowledge, and they will predict if you change this glycine to aspartate at 843, what are you going to do to the structure? What are you going to do to how it binds to other proteins? And they have all of this information, and they can make uh, they can make um, answers on how it would affect the structure of the protein based on these computations, but you never really know what's going to happen to the protein until you figure it out in your lab by creating the protein and seeing what that mutation happens, what that mutation does to the protein. So these are called computational forms of evidence. They're not real, they're virtual. They're making a prediction to the best of our knowledge, but it may not be correct. So that's why they can't be used solely to reclassify any variants. They can only be suggestive evidence. And the other types of computational data is what's called evolutionary conservation. And so if this glycine in PEX1 at position 843 is conserved through primates, through other mammals, down to yeast and down to worm, you know that that's a really important residue, and it needs to be there for some function of the protein because all of the species, that have peroxisomes have a G there. So, and if it wasn't so important for function, maybe that changes could occur over time that were variants, they were benign variants. You got another amino acid there, but it didn't change the protein. So if something is evolutionary conserved, we think that is classified as a very suggestive line of evidence that it's important. And then there's well-established functional studies that are used to classify variants that show that protein that's made from that variant actually has a deleterious effect on the protein. And we learn about this through fibroblast studies that have been, contributed by families for research. And so for many of these variants, for many of the well-studied variants in the lab over years and years of time, we've been able to show what that change does to the protein's function, and that's very strong evidence because it's the most real evidence of what's happening. So these are the criteria that I used, and but we know automatically some things that are not VUSs. So if a variant is predicted to lead to an early termination, it's considered pathogenic or likely pathogenic almost all the time. So the amino acid codon changes to a stop. We talked about that, early termination. An insertion or deletion or splice site change that changes the frame. We talked about that also. These are almost always pathogenic or likely pathogenic because the prediction that they won't yield functional protein is so strong. So usually it's not a question about that. What mainly is the question are these missense variants that haven't been seen before, and haven't been studied on a functional level in fibroblasts. So then they can only use the other levels of data, and those are not considered conclusive, but only supporting. And so there are some complicated ones also where you can actually insert or delete 3 letters and then you don't change the frame because it's a 3-letter frame, but you might have an extra or a lost amino acid. And there are what we call deeper intronic changes that are much harder to classify in the spaces as to what they do to the actual protein. So, but for the most part, variants of unknown significance are most often missense alleles that have not been seen before. So the more these get reported, the more that will erase that category of variants of unknown significance. And then, but again, this is the laboratory telling us its best interpretation of that variant. For the individual patient, if they have the clinical features of Zellweger spectrum disorder, and we know what those are in all of our children and or adults, and they have the biochemical features of Zellweger spectrum disorder, well, that used to be enough because before we knew the genetics of it, we would diagnose people based on clinical and biochemical features. The only problem is once we get to that point is that we just don't know which gene it is. Not only that we don't know the change, we don't know the gene either, but we know they have a Zellweger spectrum disorder. So you could make a diagnosis of a Zellweger spectrum disorder at the level of clinical and biochemical features. That gets a bit difficult sometimes in very mild patients who are adults because we're realizing now when they get diagnosed, sometimes they don't have any of the biochemical features. So that does become a more difficult case. But at any rate, if we move down here, so now we have clinical features, biochemical features have been identified. Okay, we know the individual has Zellweger spectrum disorder, but which gene is defective and which defect is it? So this individual has 2 PEX1 variants identified, obviously, one from each parent, and one is definitely pathogenic, the lab is saying, and the other one is a VUS. So for medical management, as I just mentioned, the patient has Zellweger spectrum disorder, the individual has Zellweger spectrum disorder. We're just not certain if this second VUS is actually the variant that's causing the disorder. So it could be, and there's just not enough data yet to actually classify it. And we just went through that, the way that that's worked through. Or it's a different variant that was just missed and wasn't picked up in this sequencing. And that has happened where uh, an initial variant became too common as there were more and more variants entered into public databases, and it was decided that that was a variant now of unknown significance, couldn't be disease causing, too common, and there was actually a linked variant upstream of the gene that isn't usually sequenced that was the disease causing variant. So these are important points, but they don't change the diagnosis of Zellweger spectrum disorder in the individual. But why are gene variants important to know? So we talked about family planning, right? So we can genotype other persons that might be interested, or genotype uh, uh, a new the next pregnancy. Also, as I mentioned before, prognosis. We know that variants that predict the absence of protein are more often more severe um, in their clinical effect. We also know now that sometimes with certain missense variants, depending on where they're located, might be associated with a milder ZSD. And specifically, I mentioned PEX 2, 10, and 12. There are missense variants in certain areas that we know are associated with milder phenotypes. And then there are some, as I mentioned earlier, there are some PEX genes in which the clinical pictures are different from others or slightly different from others. And we're just beginning to get a better idea of these differences, even though there's, many, many overlaps. But there may be some specific things that are important to know about depending on which gene it is and which type of variant it is. And so moving forward, the more we learn about gene variants and these clinical pictures, the more that we will be able to assist you in management, and you are teaching us. So the other reason why gene variants are important to know is therapy. And so as we move to this age of precision medicine and individual therapies geared to your genotype, well, we need to know the genotype. And so there's many therapies for rare diseases that are advancing now in the research lab, and we hope they'll move clinically to patients that are aimed at the root cause of the disease in rare diseases. Um, this might be, maybe this is the best type of therapy because you're getting right to the root cause. Um, but it's also understanding how that gene change and protein change ends up in the clinical picture, like retinal degeneration and hearing loss and adrenal insufficiency and liver disease and developmental delays. That's always harder. That's a harder science to figure out what are the functions of this protein and why, when it's not functioning in these different cells, are we getting this clinical picture. But if it's not functioning in these different cells, we get this clinical picture. So that's a much harder scientific question and will take much longer to figure out, but it's still very good to know about because you probably could identify a ton of other therapies if you understood that, all of those pathways. But what is being understood as the easiest thing to do at the moment, or the most direct thing to do at the moment. I should clarify. The most direct thing is to treat the root cause of these conditions, which would be to replace the gene that's not working correctly, or to precisely edit that change so you have now corrected it, or to use an RNA replacement. That's the photocopy of the DNA. So instead of changing things at the level of the nuclear DNA, we could do the same thing by providing RNA replacement for that gene or edit the RNA. And the differences are that the RNA therapies don't change the genome, so they may have less adverse effect, and all of this is being, or less potential adverse effects, and all of this is being figured out. But what's not so useful is that it has to be constantly delivered at certain intervals because your RNA is impermanent and your genes are. Those RNA photocopies are made all the time and they get degraded, so they would have to be replaced. And then there are chaperone drugs that are being developed to help missense changes or amino acid changes to help those proteins that are now not folding well fold better. So these would be directed at the root cause. And then therapies that might be independent of the gene variant could be like increasing peroxisome numbers, improving downstream systems affected by peroxisome dysfunction, like improving the biochemical functions, or if there's oxidative stress, improving oxidative stress, etc. So there are still other therapies directed towards the peroxisomes themselves and directed towards the downstream pathways. But clearly, if we are going to have a treatment aimed at the root cause, we need to know the gene change and we need to know the variant in that gene. So just moving on to how the diagnosis of Zellweger spectrum has evolved over time from earliest to the present day. That initially we had clinical symptoms and biochemical findings because we didn't know the genes. And then we started sequencing. And so the way we initially did this was just to look at the common variants. So most people got tested for PEX1 G843D and PEX1 I700 frameshift and a few other common variants. And then we enlarged that to have sequencing all of the genes that cause peroxisome disorders at once. And then we changed that to sequence all protein coding genes in your entire genome. And then we can even do more advanced sequencing where we could go outside the protein coded regions and into those introns, which have been really helpful in clarifying some missing variants. And now we can clarify missing variants even better, and we can get some idea of functional consequences, for clarifying our VUSs with RNA sequencing. And most of this, all of this up here was in patients who were symptomatic. So we kind of knew what we were looking for, or we could be targeted for what we were looking for. But now we have patients without symptoms, and now we have patients um, with different kinds of symptoms who are being picked up that we wouldn't have guessed would have a peroxisome disorder when we move to, whole exome and whole sequencing. And we also have newborns who don't have symptoms at all. So this has going to give us a whole bunch of new variants, I think, that are going to be stuck in the variant of unknown significance. But again, if there's biochemical findings, you become more assured of the diagnosis, and certainly with clinical symptoms. So, do you have a genetic diagnosis, or one gene variant but not the other, or one is a VUS? So, if you're in this situation, you can get, if you've never had a genetic diagnosis, so you older, you were in the generation that we didn't do gene testing, you can get tested if you have not done so. The newer technologies can pick up some of the missing variants if you have one but not other, as I mentioned. And then if one of the variants is classified as a VUS, it usually is reviewed by your genetics doctor every few years. That's the recommendation by the laboratory that does the test because new information is always available to reclassify. And then finally, the medical diagnosis of Zellweger spectrum disorder can be made without a gene test, as I mentioned, but knowing the genetic cause is empowering for all of this thing and enables for the individual and for the entire community and for therapy. So our genetic community is all the genes and proteins in our group. So even though I mainly spoke about PEX1, I showed you all of the proteins that are made. And so we go through this 1, 2, 3, 5, 6, 7, 10, 11, 12, 13, 14, 16, 19, and 26. And there's history behind some of these numbers. But what's nice is that it's the same in all organisms on the evolutionary scale. So fly has a PEX1 and worm has a PEX1, and it's almost the same protein. What's a little bit more complicated is why there's no 4, etc., and what happened to 17 and 18. But anyway, there are also genes that encode single peroxisomal enzymes in which changes will overlap clinically. So it may not be a PEX gene encoding a PEX protein required for assembly of the peroxisome, as I showed, but it could be one of the enzymes inside. And it turns out that sometimes defects in some of those enzymes and some of those proteins look very much like Zellweger spectrum disorder. So these are really part of our community too. And there's ACBD5, acyl-CoA oxidase 1, D bifunctional protein deficiency, Amicar, and adult Refsum's disease. And then they're not all autosomal recessive. So one of the always interesting things about learning and teaching genetics is that there are no hard rules. And so we always end up learning something that we didn't think we knew before. And so we used to say Zellweger spectrum disorder was recessive. Now we know that there are dominant or pseudo-dominant forms, really rare, but they have been diagnosed with certain changes in PEX10 and PEX6. So in this case, you're never going to find the other allele because there isn't one because it's just a single allele that causes the disease, even if the other one is normal in these unusual cases. So that's it, and this talk is open for questions.
Thank you so much, Dr. Braverman. That was fantastic and just such an important presentation. Thank you. Um, we're going to move into our Q&A portion now. As a reminder to our audience, you can use that Q&A button below the webinar player. You can also drop your questions in the chat box. If you use the Q&A button, you can also select to submit anonymously. And if you use the chat box, they're going to come directly right through to Ryan and I. And I'm going to go ahead and get us started. Um, Once a lab has a sample in hand, Dr. Braverman, how long does it usually take before families can expect results about identified variants?
Yeah. So the usual time course that we tell families is 4 to 6 weeks once it's in the lab. It can be faster than that. There are expedited sequencing reports that are requested by physicians too, if it's really important, like the infant is sick and in the nursery. So you can get faster turnaround times, like 2 or 3 weeks if that's requested. Otherwise, it's typically around 4 to 6 weeks.
Thanks so much. And we're getting some great feedback too, to let you know, just feedback already coming in through chat, not just questions. Fascinating. Thank you so much for making this user friendly, is the words that they used. Really making this user friendly information, Dr. Braverman. So thank you. We do have another question. For future newborns or growing families, how can we test the specific variants, or do we just do a whole genetic panel?
Okay, so if the child is healthy, then you know they don't have they're either a carrier or they have two normal alleles at that position. It doesn't matter, they're normal. And so usually we try to do, and this is my genetic counseling side, is that we try to say maybe it's in the interest of that person to do it when they decide they want to do it. And so if a child is healthy and there's no reason to determine whether they're a carrier or not, you wouldn't offer that test, or you would try to say that the test, the individual could determine whether they want that test when they're an adult. Um, did that answer the question?
I'm hoping so.
Okay.
They can
But
certainly write back in if they have an additional second part to that. Please feel
the
free. That was
yeah,
submitted through chat.
the siblings of the parents who are going to be carriers could get tested anytime they want, and it's up to them also. So usually, the recommendation is to see a genetic counselor and talk about it, and then you make the decision of whether you want to be tested or not. Because remember, both parents have to be carriers, not just the parent that's in the family with the carriers. And so the chances of your partner being a carrier can be given a number, even though clearly when it happens, but it's still a very low number when it's given as a chance. And then the probability of having an affected child is 1 in 4 in every pregnancy, which makes the number even lower. So once it's really important, I think, to understand this and to make a decision based on that understanding of whether you want to be carrier tested or not.
Oh, thank you. And that person did actually write back in and say they were asking specifically about parents with new pregnancies, not the actual person, and asking about certain tests. And I have that person's question, and we can probably follow up and share some information with you more about that if you'd like. We do have another question about someone who tested their child tested positive for pathogenic PEX6 variant, and they said that they were not shown to be a carrier. The next step is for their husband to see if he is a carrier. Is there more testing that their son may need to get done to confirm a diagnosis?
Okay. Well, that could be one of, remember in my last slide, I said that, we learn something new every day, and there's some forms of Zellweger spectrum disorder that are dominant. So you only need a single non-working gene where the other one can be normal. And that just happens to be PEX6 and PEX10. So maybe this is the pseudo-dominant form of PEX6. And that's very complicated to explain. I even have problems explaining it to other geneticists. But it can occur de novo in that child. So in the embryo that made that child, it occur as a new change in either the sperm or the egg, which means both parents might not be carriers, and their risk in another pregnancy would be much, much less for some de novo, or what we even call an accident when something like that happens, for the chance for that happening again. Or it could be that the father carries it, and he doesn't express the disease because of a complicated genetics that has to do with overexpression of one gene over the other. And so he may have an arrangement that doesn't overexpress, but when he passed it on to his son ha received it in an arrangement in which it overexpresses and therefore causes the disease. So it's complicated. It would be helpful to have a piece of paper and show this. But what the situation that you're explaining to me could be the pseudodominant form of PEX6. It could have occurred de novo and will not be in the father either, or it could be in the father, but he doesn't have disease because of the way the genes are expressed in him versus his child. I hope that
And
explains things.
thank you so much for taking the time. I know that that was a challenging one, especially when you don't have that paper and pen to show. So I'm happy to the person who submitted that question. We can also connect after this webinar as well. Ryan, did you have some questions too that were coming in through Q&A that you wanted to ask?
Yes. Yeah, and one that I was actually curious about because I fall under the same category. So this writer said, "We sent fibroblasts a very long time ago to your lab, like you mentioned. I was just curious if they were still being studied or if they are studied for a certain amount of time. I'm so glad we could contribute to the study of this disease in this way."
Yeah, so all of the skin cells that we have collected, it's just called a biobank. We use them all the time, and they're really, really helpful. So when we were doing, for example, drug studies in our lab to see if we could identify drugs that would improve the folding of these misfolded proteins, we took out the cell lines. We don't know who they are. They're called, number 1209, number 1206, but and we knew the genotype. So we knew we took out a cell line with a genotype, let's say PEX1 G843D over I700 frame shift. And that could have been, for example, your child's. And we would test those cell lines to see if they were covered with a drug. And we would test many cell lines. And then, if there were the end of that project would be published. And so the information would be out there for everyone to use. And then we test the cell lines, you know, for um, uh, for all sorts of other biochemical functions, for other possible changes downstream of the non-working peroxisomes. So they're used quite a bit. We have projects that we keep thinking of that we pull out fibroblasts for, or we have other bona fide researchers who are interested in learning how peroxisomes function or doing some peroxisome function testing or learning something new, and they will contact us for cell lines and we will send them. They're all anonymized. So this way, it's more than just our lab, it's the community that we hope will bring back to patients this knowledge that we learn about peroxisomes. So another idea we had once was to look for modifiers of the gene expression. So remember, we have 30,000 other genes and they're going to change gene expression, and the disease in one person is not going to look exactly like the disease in another person. And so we wanted to try to understand some of those modifiers. So we went back to the cells and looked at them, and they're a source of not only genetic material, but the RNA material and the protein as well. Um, if we do things like RNA editing, we could use the fibroblasts and see if these changes could be edited. So this just, I hope that gives you some idea of how they're really, really useful and continue to be useful, not only to us, but to, the idea is to share where we can learn. So the more people involved, the more heads, the faster we're going to learn and the more we're going to learn.
Yes, that was definitely helpful. Thank you. And thank you to those out in the audience who have participated in those efforts.
And there are, to make sure that this is all done, legitimately, there's something called a material transfer arrangement. So if another investigator wanted the cells that we banked and that you consented to allow us to bank, a material transfer arrangement has to be filled out so we know that they have a research approved protocol at their institution and exactly what they're going to do with the cells and how long they're going to keep the cells, etc. So all of that is, very quite really controlled at this time. It didn't used to be about 20 years ago, but it's all quite controlled and everything is used legitimately.
Thank you so much. You even, I think, touched on some questions that have been asked, I think, in different ways, and even before today's webinar about when you see, individuals within the same family with the same variants and how they can clinically appear, differently. I'm going to shift over to a question that we have about inheritance pattern. And bear with me because I want to make sure I get their question fully asked. They're asking about, can you explain three babies born back to back with ZSD? And they have the word stop protein, so I'm thinking that maybe they're talking about, the frame shift mutations,
Mhm.
in relation to the 25% affected, the 50% carrier. So maybe a little bit of a two-part question of, making sure that, autosomal recessive is for each pregnancy, just trying to confirm
Mhm.
that, and that's how that's possible. And then also would the difference in the variants, impact that?
Okay, but the three children have the same genotypes, right?
Yes.
They just have different clinical pictures.
They had three babies back to back diagnosed with ZSD. And they're just trying to understand that in relation to the inheritance pattern, how that's, if that's possible, how that's possible.
Right, right. So each pregnancy is like flipping the dice. You can flip three times in a row that small a, small a. And so the if you pull, but if you flip 100 times, this is going back to, statistics, basic statistics. If you flip 100 times, you probably get closer to that 25 two big A's, 50 a big A and a small a, and 25 two small A's. But when you only have a small number of children, you're flipping that dice each time, and it's just the way it comes up. So if you have, and we don't have control about over that unless we were had a pre-implantation genetic diagnosis where we can control those particular genes that we give to our to those developing embryos. But otherwise, we don't have control over it, and the two small A's can get together one, two, three times in a row just because it's a probability. And equally, you could be two parents could be carriers and you could have 10 children and none of them would be affected because you never flipped two small A's together. So I hope that enables you to understand it better. The clinical pictures are usually similar, but there can be differences. And when there are big differences in clinical pictures with the same genotype, we look for something else. And we have found it in other cases where there's another third variant, pathogenic variant, that hits the protein that's made, but doesn't work as well to knock it out. So in a case where you would have a protein that's made like G843D and a child born with a very severe clinical picture that's different from the other children, we have found in a single situation where we looked another change that occurred de novo and made that that protein absent. So that would be one example of how other changes can affect the clinical picture.
Thank you so much. And I think we have time for one more. And I think you touched on this lightly about Variants of unknown significance being reviewed by your genetic doctor every few years as new information is available. But the question that has come in is a little bit more about, is the lab or clinical team continuing automatically to look at that for that second variant? Should families reach out periodically? Is this automatically happening? How can a family be proactive about finding that second one if only one has actually been identified?
Yeah, so that's a good question. I don't think the I don't think all labs review previous reports regularly. I think it really is up to the genetics center to do that. Uh, so you'd have to speak to your geneticist to see how that worked, but you could always, yourself ask. And that's a reason to continue to see your geneticist. As one of the reasons I think, but I'm a medical geneticist, so I might be biased, is that we look at the whole patient. So we just don't look at the liver or the eyes or the hearing. And we make sure everything is being done and is being done appropriately. And we would be the person who would be responsible for really making sure that no new information has come up on that variant that might change the classification. And we would be the one who might know of the studies that could be done to determine whether that variant is actually disrupting protein function, like what research studies would be done where. So I think that the best answer to that, to be sure it's being done, is to bring it up with your geneticist and to see them on regular visits to make sure everything's being taken care of as it should be. All of the systems are being followed in a way that we know is the best in 2026, because every year we learn something new.
Isn't that true? Thank you. And I know I said that was the last one, but I think this is so important because of the natural history studies that are happening. And just we've talked today about just the importance of being able to share this information for so many reasons. When you have a family who is bereaved and they are not sure of the variants that their child had, the child has passed away from years ago. We had a few that were asking, how do we go about finding those variants so that we can provide that with the medical information they're sharing for natural history studies?
Yeah, so I think that's a difficult question because it's going to be who really is going to be the parents should be carriers, so they could have a PEX a gene panel done because we don't have to sequence all the genes. We know what disease it is, so we could just focus on those genes. But then the question is who would pay for it, with the insurance company, because sometimes they're not going to pay for looking for that answer in the parents who are healthy. But on a research basis, that could be done. And then you have, if there's any material that's left on the infant, sometimes if there was a surgery and something was saved, that is often banked in hospitals. And so if that could be retrieved, then you could look at the DNA, you could sequence the DNA of the individual themselves. But you should be able to figure it out from the parents and sequencing all the PEX genes as well.
Thank you so much. It sounds like there's a few different avenues. So hopefully that will be helpful to our bereaved families to hear. And also, I just really appreciate you being mindful of the cost involved and the differences of scenarios that can happen. Ryan, did you have any others that you wanted to try to squeeze in?
No, I don't. Thank you very much, Dr. Braverman.
You're welcome.
So if you could go ahead and move on to the next slide for me. Just want to thank everybody as we wrap up today. Dr. Braverman, just really grateful for you being here, helping us all better understand this important topic. Just all that you do for our community as well, not just today's webinar, but how much you work and you're just committed to helping individuals with peroxisomal disorders. We appreciate you. To our audience, the QR code on the left side of the screen will take you to the web page where you can learn more and get involved in the GFPD patient registry for peroxisomal disorders. There's a QR code on the right side of the screen, and that's where you can sign up for the GFP Top 3 newsletter to receive our biweekly updates, which include information about events like today's webinar, also, research opportunities, community updates, and just so much more. We also encourage you to follow us on social media and stay connected. We will be sending out links to everything shared today, including contact information so you can learn more and participate in the natural history study at McGill University. Finally, please keep an eye out for a short post-webinar survey. It should pop up actually when you exit today's webinar. Your feedback's incredibly important to us. It helps shape our future programs like this webinar today. So thank you again, everyone. We hope you have a wonderful rest of your day or evening, wherever you're joining from. Thank you.